Today's talk is entitled Overcoming Late-Stage Formulations and Manufacturing Challenges with KinetiSol Technology. My name is Ryan Muse, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speaker. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and your comments for our speaker throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit your questions as well via the comments tab. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming to those who have registered on xtalks.com. At this point, I'd like to thank Alston PX, who developed the content for this presentation. Alston PX Pharmaceutics and Manufacturing is a CDMO providing analytical and formulation development services and cGMP manufacturing for small molecule drugs. Alston PX specializes in phase-appropriate development strategies, speed to clinic and market strategies, and bioavailability enhancement for poorly soluble molecules, including their next-generation amorphous dispersion platform, KinetiSol technology. But now I'd like to introduce our speaker for today's event. Dr. Dave Miller currently serves as the Chief Scientific Officer at Alston PX, where he leads the pharmaceutics and analytical development teams and oversees the application of the KinetiSol technology. He specializes in formulation and processing of technologies for improving the oral bioavailability of insoluble small molecules. He has applied his expertise toward advancing numerous drug candidates through all stages of development, from early discovery to line extensions. But now, without further ado, let's go ahead and hand things over to our speaker for today, Dr. Dave Miller. You may begin when you're ready.
Thank you, Ryan. Thanks for that introduction, and thanks everyone for joining today. These X-Talks webinars are always fun to do, so I'm excited to be with you to talk today about overcoming late-stage formulation and manufacturing challenges using KinetiSol technology. So over the next 45 minutes, I'll walk through some of the common bottlenecks that we face in developing ASDs, especially as we move projects towards scale up and commercialization. We'll look at where conventional spray drying approaches start to break down and how KinetiSol can overcome some of those barriers. I'll share some real world examples with molecules that many of you may recognize to help ground these concepts in reality. And my goal is that by the end of the session, you'll have a clear picture of when and why KinetiSol can be a strategic enabler for late stage programs. So let's start with the basics. It's probably very basic for most of you, but let's lay the foundation here for what we're ultimately trying to achieve. These days, as formulation scientists, we're often fighting the same battle: poor solubility leading to poor bioavailability. So the amorphous solid dispersion approach is a really powerful tool to overcome this problem, where we can reduce crystalline drug particles down to the molecular scale and stabilize them in a polymer matrix. And the goals are to enhance dissolution and solubility, prevent recrystallization of the amorphous form on storage, delay precipitation in vivo, and enable tailored release profiles that align with the target product concept. When done right, ASDs give us an order of magnitude increase in apparent solubility that can enable therapies that would otherwise fail due to poor exposure. And importantly, ASDs allow us to do this in a form that's manufacturable, stable, and amenable to patient-centric solid dosage forms. So let's talk about the benefits of spray drying in early development. So, for the past 2 decades, spray drying has been the workhorse for ASDs. It's an extremely useful tool in early development because it's accessible, flexible, relatively quick to iterate. Small scale units are widely available that allow proof of concept work with minimal API consumption. It's ideal for generating binary ASDs, particularly for early pharmacokinetic evaluation in animals. And many organizations have well-established template-driven workflows for spray drying development. So in early discovery and preclinical phases, it often makes sense to start with spray drying. But we've seen the story can change dramatically once those programs move downstream. So scale up is where spray drying really starts to reveal cracks. First, these simple binary formulations that tend to dominate the SDD space, they can suffice in early phases, but can turn out to be suboptimal, due to issues like low drug loading or inefficient oral absorption that lead to things like high pill burden, amongst other issues. Also, what works at the benchtop scale doesn't always translate when you move to pilot or commercial. Spray drying systems are sensitive to parameters like feed solids concentration, mode of atomization, residence time, et cetera. And with those changes, the particle size distribution and morphology of the particles can change. That in turn can alter downstream processing and sometimes biopharmaceutical performance. And not to mention that a lot of the feed solutions for the SDD processes that we've seen lately are typically very dilute, low solids concentration. So this requires enormous volumes of solvent per kilo of SDD. And this also translates into long processing times and always substantial energy usage. And furthermore, the post-processing steps after the spray drying piece is, secondary drying, roller compaction. These add unit operations to the total manufacturing process and also can increase the mass of the dose unit. So collectively, these factors add risk, cost, complexity, and time, at a place in development when programs can least afford delays or disruptions. So let's talk about how SDD particles can change on scale up. To visualize this, you think about how small shifts in spray dryer, geometry, scale, nozzle type or performance, how those can ripple through the process. So when the mode of atomization or residence time changes, so too can particle size and density. Larger, more dense particles, for example, can lead to variations in residual solvent content, changes in internal structure. And again, those differences can cascade into variances in downstream processing and performance. And the variability isn't just a nuisance, it can lead to major manufacturing and product inconsistencies. In some cases, the product's bioavailability changes enough that you got to go through reformulation and PK bridging studies that can add significant time and cost to the program. And we put some SEM images and particle size distribution figure below from one of our partners, Boehringer Ingelheim. This is exactly the issue that they were dealing with and going from small batch to large batch supply and the drastic changes in particle size and morphology that were causing significant dosing and performance issues. So let's look at some real life commercial examples of the issues that can be encountered with the spray drying. So first we look at this Intalence, which is the spray dried form of etravirine. Intalence was approved first in 2008 as a second line therapy for HIV-1. The molecule is a BCS class 4 compound, so it's poorly soluble and poorly permeable. The commercialized spray dried dispersion is etravirine and HPMC at a 25% drug load. And because of that relatively low loading and the need for external excipients in the tableting phase of the SDD product, each of the 200 milligram tablet strength, they weigh about 1.4 grams compression weight. And the dimensions are 22 by 11 millimeters. So it's a very large tablet at the higher dose. And patients have to take 400 milligrams a day. So we take these pills, these 200 milligrams twice daily, which is not the most convenient therapy. And beyond this, the product also shows relatively high pharmacokinetic variability as well. So ultimately, the product and the process work, but it's definitely not elegant. And these large tablets are not patient-centric. So let's look at some of the manufacturing details for the spray drying process. So this process really helps to highlight the inefficiencies of solvent-based ASD manufacturing. So in order to make that etravirine HPMC dispersion, the innovator had to use 63 kilograms of dichloromethane and 7 kilograms of ethanol per kilo of API. That's just a huge ratio. And after spray drying, obviously the product requires secondary drying to remove the residual solvent, followed by roller compaction to improve density, flow, compressibility. And each of the 200 milligram tablets requires 40% external phase excipients to achieve acceptable tableting properties. And these steps are obviously at cost, complexity. The use of dichloromethane in that amount is an environmental burden potentially. And just think like this is just one API. This is just etravirine. Now multiply this across the entire pipeline of spray dried products, and you see why, as an industry, we need alternatives. So when you scale up this the Intolence product, that's where we start to see an even starker picture. So we calculated that 5 metric tons of SDD are required annually to supply the US market alone. This is based on pre-genericized prescription data between 2020 and 2021. So to produce that 5 metric tons of the Intolence dispersion, it consumes 78 metric tons of dichloromethane, 8.7 metric tons of ethanol, and 567 metric tons of nitrogen, of our total nitrogen that's recycled through the process to produce this material, and conditioned, meaning it's got to be heated to evaporate the solvent, cooled to knock out the solvent, and then reheated as it re-enters the process. So if you add that recycle of nitrogen through the process, it's a substantial mass of processed gas that's being continuously conditioned, which is a good indicator of the energy consumption. So the total processing time to produce 5 metric tons of ASD by this process is about 480 hours. So while spray drying, making this product by this process is from an engineering standpoint is an incredible accomplishment. From sustainability and cost perspective, it's very inefficient, and yet spray drying remains the template that most organizations still follow for late stage ASD manufacturing. To examine another case of SDD limitations, we look at Erleada, the tablet contains a spray dried apalutamide in a 25% drug load in HPMCAS. So apalutamide is a next generation androgen receptor inhibitor used for the treatment of prostate cancer. There's 2 dosage form options. The total daily dose is 240 milligrams. So you can either take 4 60 milligram tablets or 1 240 milligram tablet. The 240 milligram tablet weighs, again, over a gram in this high tablet mass, was such a concern for swallowability in a dysphagic population of patients that the high strengths were not approved in Japan and a few other Asian countries. So instead there, the patients have to take 4 of these 60 milligram tablets to achieve the same dose. Again, it's effective drug delivery. It enables the therapy, but at the cost of patient convenience and adherence potentially. So now, this is where KinetiSol enters the picture. We view KinetiSol as the next generation ASD technology. It's a solvent-free technology designed specifically to overcome some of these inherent constraints of spray drying and hot melt extrusion. The technology began life in 1997 as a plastics recycling innovation, which was an elegant engineering solution for dispersing incompatible polymers. A decade later, that same principle was applied to pharmaceutical formulation challenges. And since 2008, we've refined KinetiSol into a fully commercialized platform with world-class engineering and pharmaceutical science behind it. The idea is simple but powerful. We harness frictional and shear energies to instantly mix, melt, and disperse the API in the polymer without the use of solvents or external heat. And what results is a molecularly uniform amorphous dispersion that's produced in seconds as opposed to minutes, and without the uses of solvent or applied heat, as I mentioned. So mechanically, I have a video here that I'll show that you all can watch while I talk through this slide. Mechanically, KinetiSol operates, very, very different from either spray drying or extrusion. The solid blend of drug and matrix excipients is charged into the high-speed mixing chamber. As the blades rotate, they generate first friction in the powder bed and then shear that raises the temperature of the mass just enough to form a homogeneous melt and dissolve the crystalline drug substance. Once the dispersion is complete, the material is ejected and rapidly quenched, locking in the amorphous structure and forming an amorphous disc that you can see depicted in this video in just a moment. The entire process typically takes only 20 seconds from start to finish. And because no solvent is used, there's no need for secondary drying. And after milling, KinetiSol particles are dense, freely flowing, directly compressible. There's no need for densification through roller compaction or other forms of granulation. And very importantly, the equipment has a small footprint and it's mobile. It can be installed in any typical GMP suite. It can be pulled in and out of those suites, and there's no special utilities, just chilled water and electricity. So now I want to take a minute to talk about the formulation design space that's enabled by KinetiSol. And it's one of the biggest advantages of the technology and the freedom that it gives formulators to design amorphous solid dispersion specifically to the needs of their compounds. So with spray drying, you're often limited by common solvent solubility, feed solution viscosity, etc. when you're trying to design a formulation. And with melt extrusion, you're limited by the thermal stability of excipients. Can they withstand the heat of the process, and also the rheology, the melt rheology of the polymers? There's a certain viscosity range that's amenable to extrusion, and beyond that range, the formulations simply don't process. So KinetiSol sidesteps all of these constraints. Because the exposure time is short, even thermally labile compounds can be processed safely, and there's no issue solubilizing high melting point compounds into these polymer melts. So we can process these high melt point compounds almost independently of their melting temperature, several, up to 100 degrees below the melting temperature, in fact. And with KinetiSol, you can work with polymers that are non-thermoplastic, highly viscous, are the ones that are insoluble in organic solvents. And to these, you can add virtually any other adjuvant material like lipids, surfactants, stabilizers, what have you, in any combination. So this opens up a much wider design space and enables a formulator to bring, not only introduce more APIs into the amorphous dispersion domain, but also to tailor the ASD matrices to meet the specific challenge of your molecules. So in short, with KinetiSol lets the science dictate the formulation design, not the process limitations. So very powerful in terms of formulation design. So let's look at a real case here where KinetiSol transformed a late stage program. So a client approached us recently with an SDD formulation from their phase 1-2 studies. Their product showed suboptimal bioavailability, high pill burden, and manufacturing complexity. So using KinetiSol, we were able to reformulate the ASD and using that wider design space and deliver a 5-fold increase in AUC in a primate model. And as a recent follow-up, it was confirmed that this enhancement did translate well to humans, and consequently, we're now planning for phase 3 supply. But the improved performance also allowed the dose to be reduced and the regimen to be reduced to a single tablet as opposed to multiple tablets. In terms of timing to bring this about, it only took 4 weeks for us to deliver prototypes for animal PK, and once confirmed in the primate model, we initiated clinical trial material, and the product was delivered in 4 months. So rapid turnaround from prototyping to manufacturing with KinetiSol. These manufacturing timelines are very short. And just as important, as I talked, we reduced process complexity by eliminating solvents and reducing the manufacturing time by about 2/3, and obviously simplifying the scale-up path from prototype to initial PK supply to now late-stage supply. So this is a really powerful example of how a technology transition can rescue a late-stage asset and set it up for long-term success. So now we want to talk about the tableting, the process flow, and I'll show you how the process flow compares between spray drying and KinetiSol. So here's the 2 process trains side by side. So as most of you know, the spray drying route involves multiple downstream optimization operations, secondary drying, roller compaction, blending, and finally tableting. And each of those unit operations adds time and handling, and with roller compaction, additional excipient dilution. By contrast, the KinetiSol output goes straight into milling, then blending, and directly to tableting. So there's no drying, roller compaction, and there's less excipient dilution. So the overall process is simpler and faster, much easier to scale consistently, and in many cases reduces tablet size and overall pill burden. So think of this as sort of streamlining the entire manufacturing line into a very compact, energy efficient module. So a little bit more, tableting insights with KinetiSol. So KinetiSol produces, as I said, highly compressible porous particles that really behave beautifully in direct compression. So the micropores that are created during the rapid melt quench step of the KinetiSol process, these pores become natural site for particle deformation and fracture under compression. So the result is good tablet tensile strength with rapid disintegration and minimal excipient dilution in the addition of tableting excipient. So we can often achieve very high ASD loadings per tablet, 75% or higher. So that obviously translates to less excipients in the final product and reduced pill burden. And in practice, we can, in many instances, have examples where we've reduced a multi-tablet approach to a single tablet solution in converting from spray drying or HME to KinetiSol. So, and this is a case where physics, it's not just chemistry that's working in our favor. The actual mechanical properties of our particles lend themselves better to tableting. So the tables below, they illustrate this benefit really well. So these are from our phase 3 asset, D-ferrosirox. So this was a case where KinetiSol was able to generate a 50% drug-loaded ASD using a combination of polymers and ultimately 80% ASD load in the final tablet to arrive at a 360 milligram unit dose with a 900 milligram compression weight. So that's 40% API per tablet, which is dramatic for an amorphous dispersion dosage form. So this tablet was essential to success for this therapy because it's very high dose iron chelation product. And it really illustrates how we were able to leverage all the unique attributes of KinetiSol to meet patient needs in this space. Let's talk about how KinetiSol improves manufacturability. From an operations standpoint, KinetiSol really is a game changer. A single KinetiSol compounder can replace a PSD4 spray dryer and all the associated solvent handling and process gas conditioning systems. It's portable, modular. You can install it in standard GMP suites without any special infrastructure. And because the utilities are standards, the site integration is very straightforward. And the elimination of solvents also removes major regulatory and safety burdens. No need to worry about volatile organic carbon emissions or solvent recovery or nitrogen inerting of the process. And the net effect is a faster technology transfer, lower facility cost, smaller environmental footprint. And in the environment we're in now, everything's moving towards greener manufacturing, and these benefits really are starting to matter to regulators and sponsors alike. So let's talk about how KinetiSol is managed through the different phases of development. And that's another advantage of the platform is how smoothly it supports each development phase. So at the feasibility stage, the focus is on rapid prototyping and screening of those prototypes. So the KinetiSol formulator, the small unit in the top right, really excels here because it can produce dozens of formulations in a single day with relatively low API consumption. So during clinical scale-up, reproducibility and scalability are really key. So the process parameters translate linear from grams to kilograms to tons, with the KinetiSol formulator moving to the compounder. So once at the manufacturing scale, that same piece of equipment can serve from early clinical stages throughout commercial production by simply running the process longer or operating compounders in parallel. So that continuity reduces tech transfer risk and accelerates timelines across the development cycle. So at commercial scale, the objective really shifts to efficiency, reliability, and cost control. And again, this is a place where KinetiSol pays off big, as you'll see in the next slide. So looking at the footprint of KinetiSol relative to spray drying, so the visual contrast is pretty striking. A spray drying system capable of commercial output fills an entire building bay and complete with solvent tanks, condenser scrubbers, nitrogen, all the associated utilities and operations. And there are multiple scale-up events along the way and going from prototyping to commercialization that create risk for variance in particle properties that impact downstream processing and performance, as we've already discussed. So alternatively, with KinetiSol, there's only a single scale change from prototyping to manufacturing. So a KinetiSol line delivering the equivalent throughput of a PSD4 occupies a fraction of the space. And this smaller footprint allows for installation within any standard GMP facilities without structural modifications. It also enables regional manufacturing or distributed production models that would be highly impractical with spray drying. So in an industry where capital efficiency is important, that spatial and operational compactness is a real strategic asset to most programs. So now I want to go back, now that we've set the stage, the challenges with spray drying, some of the benefits of KinetiSol, go back to some of the examples, starting with the earlier intolents. So recall the intolant story. And now early in our company's history, we undertook a development effort to generate a product bioequivalent to intolants using KinetiSol. Now remember, intolants relied on largely dichloromethane-based spray drying to create an STD of etravirine and HPMC. So in converting this to a thermal process, there were a lot of challenges. First, the compound is high melt at 270 C. It's very thermally labile. It actually degrades on melt, maybe even slightly before melt. It's highly insoluble, solubility about 1 microgram per mil. It's poorly permeable. As I said, it's a BCS4. And the spray dry dispersion was formulated with HPMC, which is a minimally thermoplastic polymer. So very challenging to translate this formulation to a thermal process. And so duplicating the STD with those challenges seemed impossible. So this slide illustrates the complete development and manufacturing journey for the KinetiSol etravirine that I call KATV. What you're seeing here is the formulation concept being developed on the gram scale and proceeding linear through pilot and into full registration scale manufacturing. So on the left, we start with the development scale KinetiSol formulator. This is the R&D system we use to make and test early prototypes to determine our lead formulation through rigorous in vitro and in vivo PK testing. And on the right is the commercial KinetiSol compounder. This is the scaled production unit that's capable of continuous operation from kilograms to metric tons. And this is where we made pilot human PK batches in the development of KATV, and then took all the way through registration scale. And the difference is manufacturing time. So pilot scale batches require a couple of hours, registration scale batches, a couple of shifts. So the center diagram between the two pieces of equipment shows how the process maps across both modes. So the process is the same. You blend, charge, compound, quench, mill. So it's the same sequence, but it's batch mode for development, and then it becomes automated and continuous for production. And the linear translation of the process parameters is what really enables reliable scale up. The registration manufacturing PAT control chart shown here on the lower right shows the process uniformity, the tight control, minimal variability, and reproducibility across thousands of KinetiSol outputs in a batch. Altogether, this slide really captures the scalability and consistency of the KinetiSol platform, going from grams to kilograms to metric tons, all while maintaining identical performance and product quality to intolants. So we took this product into a pivotal human BE study in the fasted state, and we confirmed that what we saw in preclinical in vitro and in vivo studies in this pilot human PK study. So 36 subjects received KinetiSol formulation and the intolants reference in a crossover design. And as you can see, the PK profiles were superimposable, like virtually identical in terms of Cmax and AUC in the fasted state, which the FDA really considers the pure absorption state. So we were able to exactly match the performance of spray drying in this prandial state. So, and importantly, it was noticed as well that the variability between the subjects was lower for the KinetiSol product. That's one of the issues with intolants being a highly variable product. So that really indicates tighter control over dispersion, homogeneity, and release kinetics achieved with KinetiSol. So achieving full bioequivalence for this challenging compound and ASD formulation that we thought was nearly impossible to thermally process is a really powerful statement about the capability of KinetiSol. And it also underscores that a greener, simpler manufacturing process doesn't mean that we have to compromise on therapeutic performance. And in fact, KinetiSol often does better than spray drying. It's only in this context of bioequivalence where we were aiming to be the same. We're often better. So let's revisit the intolants manufacturing now, but this time with KinetiSol as the reference point. So this table really directly compares the commercial spray drying process with KinetiSol for producing the same 5 metric ton batch of etrovirine dispersion. So again, on the spray drying side, to produce, just as a reminder, to produce 5 metric tons of dispersion required 78 metric tons of dichloromethane and 8.7 metric tons of ethanol. And once again, just to reiterate, when you consider the recycling of nitrogen gas through the process over the duration of manufacturing, the total conditioned process gas mass is over 560 metric tons. So in total, it takes 91 metric tons of liquid and over 600 metric tons of total process mass to produce 5 tons of usable dispersion. I mean, it's just a highly inefficient manufacturing process. So now when we look at the KinetiSol column, it tells a completely different story. So to produce the same 5 tons of product, we do it without solvent, with no nitrogen, and nearly 100% mass that goes into the process is converted to product. So we're not having to process a bunch of additional mass. It's focusing only on the API and the excipients coming in and exiting the process. So likewise, the processing time drops from 479 hours with spray drying down to about 250 hours with KinetiSol. So nearly 50% reduction in total ASD processing time. And once you have the KinetiSol material, it's, as I said, it's ready for direct compression. There's no residual solvents to dry off. There's no need to densify through granulation, and it only requires minimal external excipients to convert to a tablet with good disintegration and tensile strength. And obviously, from an environmental standpoint, we go from hundreds of tons of solvent to none, which is highly ideal. And from an economic standpoint, we're cutting process time, material use, and energy by a substantial margin. So when you take all of these changes together, these make KinetiSol both an economically and environmentally very friendly process, and far more sustainable for commercial manufacturing. So the bottom line, this slide really brings the message home. When you look at spray drying versus KinetiSol side by side, it's a really stark contrast. And the question is, which would you rather have? So on the left, looking at solvent consumption, we see it's, once again, not to beat this drum over and over again, but it takes tens of thousands of kilograms of solvent for spray drying, none for KinetiSol. And it just eliminates a lot of complexity and risk in terms of supply chain dependency and and just solvent handling issues, the potential future risk of regulatory constraints on the use of solvents. It just eliminates that uncertainty entirely. So in the center graph, you see the total process mass, like I said, everything that needs to move through the system to make the amount of final product. As we've already mentioned, takes hundreds of tons of material by spray drying. In KinetiSol, operates with only the active and the excipient materials themselves. So total process mass through KinetiSol is a fraction of what's done in spray drying, and that process mass is really indicative of the energy input that's required. Once again, when you think about heating, cooling, and reheating process gas, as well as handling of the solids downstream, the energy input to achieve the product is dramatically more with spray drying than in KinetiSol. And once again, in terms of total process time, KinetiSol cuts that down by more than a half. So, and when you put all of these improvements together, you deliver a substantial, we're estimating 30 to 50% reduction in cost of goods, all while dramatically reducing environmental footprint, which is becoming more and more important. I know we're hearing from our European clients that the move away from spray drying is an imperative. They're anticipating some significant regulatory restrictions on the use of solvent in drug product processing, and are looking to get ahead of this issue as soon as possible by pivoting to other ASD platforms like KinetiSol. So it's a real benefit now, where I think maybe 10, 20 years ago, solvent usage wasn't such an issue. It's really becoming a significant risk with spray drying now, and it's important to look at alternatives. So finally, yeah, revisiting which would you rather have? I think the answer is fairly obvious, looking at these variables. So on this last slide, we have a video here of our manufacturing compounder that I'll play while I once again summarize the benefits, but allow you all to visualize the KinetiSol compounder in operation. So yeah, while you watch that, I'll summarize the key benefits of KinetiSol. So once again, the expanded formulation design space allows us to maximize ASD performance and bioavailability. We can often reduce pill burden through improving drug loading. And the way that we improve drug loading is often to utilize a combination of polymers so that we don't have to compromise performance for drug load. We can utilize one polymer to maximize drug load and another to ensure delayed precipitation in vivo. So the ability to combine polymers and dense particle morphology is really key to maximizing performance and increasing drug load, reducing pill burden. We talked about minimizing the risk in scale up from R&D to GMP. Again, there's only one step change in scale and going from development to manufacturing, and that's a linear scale factor. So it's very straightforward. So again, I can't say enough about eliminating the burden of organic solvents on processing and all the complexity and cost that goes along with that. It's completely eliminated using a thermal process like KinetiSol. And then, reducing processing time and energy consumption, just, not only environmentally friendly, but reducing cost of goods towards the final product. And once again, the portability of the equipment, the flexibility, it can be installed in any GMP suite with standard utilities. So very simple, can roll in and out of manufacturing suites, doesn't need to be in a dedicated suite. You can be tableting in a room one day and doing KinetiSol in that same room the next, which is that portability is very attractive in terms of flexibility. So, I think with KinetiSol, these improvements aren't just sort of incremental. It really does represent a step change in how we approach amorphous solid dispersion design and manufacturing. And for late stage programs that are struggling with manufacturability, KinetiSol isn't just an alternative. It's an exit ramp off of complexity and other challenges that they face in dealing with large scale spray drying. Just as a final reminder, the take-home message, all of this combined equates to KinetiSol providing a substantial reduction in the cost of goods for your product. So in terms of economics of ASDs, it's, and the breadth in which it can be applied to APIs and polymers, it's by far the most economical and flexible process. With that, I'll wrap up. Once again, I just want to thank you all for joining and for your attention. I hope this session has given you a clearer picture of how KinetiSol can address some of the toughest late-stage formulation and manufacturing challenges. And whether you're working with high melt compounds or solvent-incompatible systems, or simply trying to, simplify your manufacturing process, there is a better path forward. So please don't hesitate to reach out to us if you'd like to explore potential applications or collaborations. Of course, you can always visit our website at austinpx.com. Tons of information about KinetiSol and all of our offerings, as well as virtual lab tours, written content, papers, white papers, blogs. It's a really great website, and you can always connect with us on LinkedIn as well. I'd love to continue the conversation. And so with that, I'll say thank you once again, and I look forward to your questions during the Q&A session.
Well, thank you very much for that very insightful presentation. As we move into the Q&A session, I'd like to remind the audience to keep sending their questions or comments right now for the questions window. And for those of you watching on LinkedIn Live events, feel free to post your questions in the comments sections and we'll take a look at them there. Now, I've already received some questions from the audience, though, so we'll get ourselves started with those. The very first question I have here for you just wants to know, are you able to use pH-sensitive polymers?
Yes, definitely. In some other presentations that we've done before on X Talks and other places, we kind of list out our hierarchy, not necessarily our hierarchy, but our whole full menu of polymers. So basically with KinetiSol, we can process, any polymer. We like to stick to those that are compendial and generally regarded as safe. But yeah, there's really no restriction in the pharmaceutical polymer space to what we can use. So pH sensitive polymers like HPMCAS, the methacrylates like Orgegot L155, L100, S100, and other enteric polymers. We screen those routinely through KinetiSol. Probably a majority of the ASDs that we've put into preclinical and clinical development are formulated with anionic polymers. We can also use Orgegot EPO as well. That's a component in some formulations, a cationic polymer. But yeah, routinely use pH-sensitive polymers as well as neutral polymers. We're not limited by thermoplasticity, thermal sensitivity, or viscosity. And then, like I said, I can't overstress the fact that you can combine materials as well. So as we do formulations development, we often find we do binary screening, and you'll identify that for a given molecule, one polymer has a certain benefit while it lacks in another area, while another polymer has the exact opposite properties. So you combine those two and achieve synergy, like I mentioned before, high drug load without compromising performance. A good solubilizer mixed in with a good parachute. So you can achieve high drug load without rapid precipitation in vivo. So yeah, good question.
Perfect. Thank you so much for answering that one. The next question here is from someone who's just curious if you can talk about the energy consumption for KinetiSol.
Yeah, we estimate, I think it's probably a conservative estimate that our energy consumption is probably, we say 50% less than spray drying, but I think it's less than that. So, we've got a 50 horsepower motor that's really driving the shaft and the mixing elements, and it's not running at a high speed continuously. The system sort of pulses. So it's at idle speed, and then the RPMs ramp up and ramp back down within a period of 10 seconds or so. So really all you're consuming in terms of energy is electricity to the motor, which again isn't at high RPMs for the duration of manufacturing. It sort of pulses, so it's minimized in that regard. And then we use chilled water flow to cool the chamber and the quenching system for continuous production. So there is some electricity to refrigerate the water stream. But yeah, when you consider, like I said before, in the case of etrovirine, having to condition over the course of production 560 metric tons of nitrogen, heating it up to the inlet process temperature, cooling it down to knock out the solvent, and then reheating it to the inlet, and doing that over and over again, it's dramatically more energy consumption than what I just described for KinetiSol. Good question.
Excellent. Yes, thank you. Now, the next question here is wondering, for which molecules is it not recommended to go with KinetiSol? And further, what are you thinking the disadvantages is of the process?
Yeah, so in the Venn diagram that I showed early in the process, you'll see like the formulation design space for KinetiSol is very wide, and it overlaps. It completely encircles HME, and it almost completely encircles spray drying. But there's a sliver outside of spray drying where it's outside of the KinetiSol processing space. And what that represents are molecules that are completely thermally unstable. So as they transition from a stable crystalline form into an amorphous form, they have very low heat tolerance, even for brief durations like we have with KinetiSol. So molecules that will degrade extensively within seconds of being in a non-crystalline state at elevated temperature. So we've encountered over the course of, we're approaching a couple hundred molecules now. We've encountered a handful of those that catastrophically degrade as you convert them to an amorphous form and at elevated temperature, and for which the degradation isn't at all accelerated by the polymer, just thermal degradation of the compound structure. So for those are not amenable to thermal processing of any kind, and those are ones where solvent-based approaches are the only path forward. So that's a good question. And then the question about the key limitation of KinetiSol. So I mean, obviously, so at this stage we've been developing drug products in clinical stages for well over a decade. We've worked on multiple late-stage assets, had a couple of our own in pipeline, worked with clients with late-stage assets, and NCE attrition is really challenging for all of us in this industry, but particularly for a novel technology, because it's really attaching the technology to a compound that can make it to commercialization has been a challenge. So we've been very close to the finish line on a number of occasions, and in fact now that we're a full service CDMO and KinetiSol no longer bears a royalty and is very openly available to the public, we have a lot more opportunity in late stage clinical design, in late stage clinical development. But so we're right there at the finish line of commercialization, but we haven't achieved commercialization yet, and that does put us at a disadvantage relative to other technologies. But that's quickly changing. I think within the next couple of years, that's a risk that we'll no longer be facing. And like I say, for anyone considering KinetiSol now, if you're worried about your molecule, I wouldn't worry, because by the time it becomes an issue for your compound, we'll have already commercialized the process with a couple of others. So anyway, those are both good questions.
Very good. Thank you for that answer. The next question here is wondering if you can just clarify whether the technology is proprietary or protected by any licensing agreements.
So yeah, it is. So the KinetiSol process, we've got an IP portfolio around the equipment, some pharmaceutical applications, composition matter on specific drugs. But when you partner with us, those patents are provided with a royalty-free license. So it no longer bears, the technology doesn't bear a royalty, but you do benefit from that IP protection in using the technology. So as our clients put their molecules on our equipment and carry it through development, they have the composition of matter IP specific to their drug is theirs, method of use, etc. And then any sort of foundational KinetiSol IP goes along with that in a royalty-free license. So the drug product that we develop with our clients tends to have very strong IP position, beyond just the molecule itself. And let's talk about patents specifically, but also sort of trade secret, know-how as it comes to access to the equipment and how to operate the equipment. So it's different than spray drying, where it's a commodity technology, and if you develop a spray dried formulation, it could be developed anywhere with a spray dryer. With KinetiSol, folks that were looking to duplicate your product would have to have access to the technology, which they don't. So yeah, so basically you get, in summary, you get tremendous intellectual property protection, as well as an improved manufacturing process and formulation, and that's free of charge. With it comes with the price of the development.
All right, perfect. Thank you so much for answering that one as well. Moving on here, I've got a question from someone curious about what are the scale-up capabilities for commercial manufacturing, and how many sites are available for manufacturing using this?
Yeah, very good question. So early development through phase 2 is handled entirely in-house here at Austin Pharmaceutics, where we have the dedicated development and GMP suites equipped with both the KinetiSol formulator and the KinetiSol compounder systems. And once a program progresses beyond phase 2, we would execute a technology transfer along one of 3 possible paths, depending on client preference and commercial strategy. So we could transfer to one of our established CMO partners. Or to a mutually agreed CMO site could be set up as selected by the client, or in a lot of cases directly to our client's own facility when that's a preference. So the idea is that we aim to be flexible with the arrangements for late stage manufacturing and commercialization with our partners. And in our along that first path, our past CMO partners and currently Microsize in Pennsylvania is a very close ally that with tons of expertise in providing drug product intermediate. So they're one of our more preferred partners for producing KinetiSol intermediate that would ultimately be transferred to another site for finish of the final dose. And then we've used 2 other sites in the greater New York area, one of which had a dedicated KinetiSol suite And there are other strategic partnerships in development with some other major players in different parts of the U.S. But we want this network to be flexible to ensure that sponsors can move seamlessly from feasibility to commercial supply without redevelopment, and also have redundancy as well. So that's a very good question.
Great, thank you very much for that answer. I've got a big one here for you. Someone says that there's recently been an increase in the use of lipid-based excipients to enhance solubility and permeation in solid oral dosage forms. They're wondering, what is the compatibility of these excipients with KinetiSol processing? And if certain excipients key to product performance are incompatible with KinetiSol processing, can they be added to the KinetiSol particles in a second blending operation prior to compression?
Yeah, that's a very good point. And it's something that we've been seeing as well, sort of an uptick in interest in lipids. There's always been a lot of interest in lipids, but more specifically, their synergies in amorphous dispersions. And I think the KinetiSol process is perfectly suited to generating sort of ASD lipid hybrids. And the reason being is that the melt mixing mechanism of KinetiSol is highly turbulent. So the drug polymer lipid mixture is mixed very rapidly and turbulently. And then as it's ejected, you saw in the animation, it's immediately quenched. So it's turbulent, it's well mixed, and then it's frozen in place in that quenching operation. And so it doesn't give time for phase separation of the lipid excipient from the polymer matrix. So it makes a homogeneous mix. And we did some research very early on in KinetiSol development with an excipient company, where we established that we were able to make homogeneous polymer lipid mixtures using KinetiSol up to 40% lipid content. And we contrasted this with extrusion, and what we found is when you try to make these same combinations with extrusion, as you escalate the lipid concentration, you'll see right at the die of the extruder phase separation. You'll have a lipid stream dripping down the front of the die, while your polymer-rich strand comes out above it. So that sort of laminar flow towards the end of a barrel leads to phase separation by viscosity and limits the ability to co-mix viscous excipients like polymers with low viscosity excipients like lipids. But again, just to reiterate, KinetiSol doesn't have this issue because it's highly turbulent mixing that's rapidly frozen in place through a quenching operation. And so yeah, we think KinetiSol is an ideal platform for making these polymer lipid composites, and the synergies in terms of performance, like you said, are starting to emerge, where lipids are improving, solubility, permeability, just overall bioavailability for really challenging molecules. So yeah, that's a very good question. And then there was a second part, Ryan, and I'm forgetting it at the moment. Can you remind me?
They're wondering if certain excipients key to the product performance are incompatible with KinetiSol processing, can they be added to the KinetiSol particles in a second blending operation prior to compression?
Yeah, that's a really good question. Yes, the answer is yes. This is a strategy we've employed with different formulations, adding performance enhancers extra granularly. Most often this is surfactant materials. We've also added cyclodextrins extra granularly as well, to great effect. In terms of, so the driver for doing that can be different. The incompatibility issue tends to arise most often with something like sodium lauryl sulfate. It just tends to be very reactive. A lot of drugs are incompatible with SLS in the amorphous state, especially at elevated temperature. And SLS, although it's a pretty aggressive surfactant, it's not preferred in oral formulations, but sometimes it really is the key towards achieving target performance. So in those cases, adding SLS externally to the granules has been a very powerful approach. So anytime a surfactant material tends to be reactive with the drug, that's a strategy that we've utilized. But also not just limiting it to surfactants, but even the use of polymers like Soluplus, for example, that tend to have a bioavailability enhancing effect in some cases, but the low TG of the polymer can limit drug load. We've also evaluated with success using Soluplus external to the ASD granules. So yeah, this is a somewhat common approach that we've used. So yeah, very good questions.
Thank you again for that answer. I think you covered a lot of it for him. Another question I've got that's come in for you is wondering what specific triggers would you say could prompt a late stage pivot to KinetiSol?
Yeah, that's a good question. So there's a couple of different triggers here. The first set usually arises at the formulation level, where and these are very urgent, where performance limitations become too great to ignore. So when a formulation that was working well in early development or good enough, basically that's really what happens. I think a lot of companies just need to get safety and an early efficacy proof of concept. They go in with a good enough binary dispersion, and then when you transition to late stage, these spray dried dispersions begin to show variable or undesirable pharmacokinetic profiles. Maybe it's poor bioavailability that's driving up dose and pill burden. So these are things we've seen on a number of occasions. And like I said, this just traces back to fundamental formulation constraints for spray drying. They're often binary systems that either they don't achieve sufficient amorphous stability, or at least rapid precipitation in vivo, or there's inconsistencies in release. So when early clinical data are acceptable, but these limitations really become magnified as you go into pivotal studies and move towards commercialization. So that's where the emergency to explore KinetiSol to reformulate the spray dried dispersion into something that meets the performance targets. That's really one of the more urgent triggers, is when the drug product performance isn't matching with the clinical teams and the commercial groups expectations. So the second group of triggers really stems from manufacturing inefficiencies that are inherent to spray drying. So a lot of the spray dried processes that we see these days have very low solids load in the feed, under 10%, under 5%, sometimes less than 3% total solids, and that makes spray drying just highly inefficient, volumetrically. And as you dramatically increase the amount of solvent and gas throughput that's required per kilo of SDD. So as the batch sizes grow, these low solids concentrations lead to large scale throughput bottlenecks, and not to mention, environmental and safety burdens. So that's been a driver towards converting to KinetiSol, is just looking at the realities of manufacturing a spray dried product with very low solids low in the feed. The other thing is solvent selection, another key constraint. So we see spray dried processes that are based on solvents like THF, and I already showed dichloromethane a couple of times in this presentation. They're required to dissolve the API in the polymer. So at a commercial scale, those solvents become a liability, and they're expensive, and THF is got explosion hazards. DCM is regulated. I think it's going to be more highly regulated in the future. So it's difficult to justify long-term manufacturing with those types of solvents. So yeah, you combine all of these challenges, and that's what typically triggers serious evaluation of an alternative technology. And KinetiSol becomes a logical pivot point because once again, we can improve the product, we can dramatically simplify the process and increase efficiencies. So anyway, that's where in these scenarios, KinetiSol provides a technical rescue and economic optimization for late stage programs. So yeah, very good question.
Perfect. Thank you so much for that answer and for all of the answers today, but we have reached the end of our time for the webinar. Now, if we couldn't attend to your questions, know that the team at Austin PX will follow up with you, or if you have further questions, you can direct them to the email address that's up on the screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it helps us to improve our webinars. Now, additionally, I've shared a link to view the recording of today's event in the chat box, which you can also share with your colleagues so they may register for the recording here as well. For those watching on LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and gain access to features such as viewing the recording and more. Now, please join me once more in thanking our speaker for his wonderful time here today. We hope that you found the webinar informative. Have a great day, everyone, and thank you for coming.